So, good afternoon. So, welcome back. So, in the previous sessions we have discussed that anatomy of the digestive system. You know the digestive system which is composed of the alimentary canal or gastrointestinal tract and the digestive glands. Okay, in the previous classes we have discussed the anatomy, that structure of the digestive system. Okay, so actually, in this session, so we are going to discuss that physiology of digestion, that process of digestion. Okay, the digesting the food, the digestion of the food, and what is the role played by the enzyme. So, what are the role of the enzymes and process of digestion we are going to discuss, right. So, you know already that digestion, it is the process of that conversion of the complex into simpler, into an absorbable form. Okay, absorbable and assimilable form is called digestion. So, already you know, and this already two types of digestion. So, it is accomplished by the chemical and the mechanical digestion. So, you know that chemical, that complex substances like starch is converted into glucose, that is chemical digestion. Protein are converted into amino acids, lipids are converted into fatty acid and glycerol. Okay, that is called, it is chemical digestion. And mechanical digestion, when chewing the food, so that break the larger particles into simpler, that is smaller particles, that is mechanically that breaking. Okay, just like the breaking the large stone into pieces, it is a physical change, it is a mechanical change. So, that digestion, that both digestion takes place, namely the mechanical digestion as well as the chemical digestion, right. Okay, what is digestion? It is a 2 marks. Define digestion. It is a process by which that conversion of the solid food into absorbable and assimilable form is called digestion. So, which is accomplished by the mechanical and the chemical, the two types of digestion, mechanical and so okay. So, now we are going to discuss the process of digestion. When that food is introduced into the buccal cavity. So, buccal cavity through the mouth, that food when introduced, so at that moment, when that is after the introduction of the food, that process of digestion starts. At that immediately after that second, that process it started. Yeah, you started chewing the food. When chewing, that food is break into pieces, that is mechanical digestion. Also chemical digestion start. Okay, both started when the food is introduced into the buccal cavity. So, one by one we are going to discuss. So, one by one. First, what is the process of digestion? What are the changes takes place in the buccal cavity? That first phase, listen. So, that process of digestion and role of enzymes. Without the enzyme, no chemical digestion. So, when the food introduced into that buccal cavity, so that buccal cavity, that is, you know, the 3 pairs of salivary gland, that salivary gland started that secreting the saliva, So, that salivary secretion depends the smell. When you feel that smell, if you sense the smell, okay, that started saliva, that started secreting the saliva. That is why that food is, we add some spices to add smell, because that induce the secretion of the saliva, okay, by the salivary gland. So, that smell of the food when seeing, okay, that sight, taste, okay, and also that mechanical, then after the that food is introduced into that buccal cavity when chewing. So that what is that? That mechanical, that contact, okay, food that contact with what is that, palate and gums, that induces the stimulation, okay, of the salivary secretion. It stimulates of that salivary gland, so that it is a reflex action. Okay, it is a reflex. Okay, and so that because of these are what is that, stimuli, that smell is one of the stimuli, sight, taste, all. Okay, so that as a result, that reflex action that is started, as a result, that saliva starts secreting saliva. So that is saliva. What are the composition of saliva? Next, what is the composition of saliva? It contains water, electrolyte like sodium, potassium, that chloride and bicarbonates. Okay, water, electrolytes, and the enzyme. The enzyme found in the saliva is salivary amylase. That enzyme found in the saliva is salivary amylase or ptyalin. So ptyalin or salivary amylase. Also the lysozyme. It is an antibacterial agent. Lysosome. And a mucus which lubricate the food. When chewing, that mucus mixes with that food, that lubricate. When swallowing, that freely moves along the esophagus and reaches the stomach. Okay, அதான் பாருங்க, நம்ம டக்குன்னு சாப்பிட்டா என்ன ஆகும், தொண்டை அடைச்சிக்கும், அது choke ஆயிடும். Okay, so that mucus that lubricate, right? So come on, we can sum up. When seeing the food, that smell and that mechanical contact, okay, so that triggers a reflex action. So as a result, that salivary gland produces saliva. That saliva that contains the water, electrolytes, enzymes, okay, salivary amylase and the antibacterial agent lysozyme, not some. It is cell organelle, lysosome. It's a lysozyme and a mucus, a lubricating substance. Okay, that's all. And this is mainly that salivary amylase only, it is going to act upon the food and so digest, right? And when the food is introduced, the salivary gland secretes saliva and we have started chewing, that is called mastigation. Okay, we started chewing. Started chewing the food, so that is called mastigation. So at that time of the chewing or mastigation, that large food particles are break into simpler, that mechanical digestion. Okay, and okay, that what is that, mucus? Already I said. Before that, what are that enzymatic action? Enzymatic action, listen. So that salivary amylase present in the saliva, so that act upon that starch and convert the starch into disaccharide maltose. Starch வந்து என்ன பண்ணும், maltose convert பண்ணும். Okay, that starch is hydrolyzed. Hydrolyzed means it is digested. So by the action of the salivary amylase and convert into the disaccharide maltose. Starch is the polysaccharide. Okay, polysaccharide, and that maltose is the disaccharide. So that the polysaccharide starch is hydrolyzed. Hydrolyzed is nothing but it is digested into the disaccharide maltose by the action of the salivary amylase. When chewing, that saliva mixes with food and the start digesting. Listen, when it's chewing, when the mouth it is remain in the, that is the food remain in the buccal cavity, so 30% of the starch is, 30% of the starch is digested. That starch is converted into maltose by the action of the salivary amylase. By the action of salivary amylase, and so other that mucus that softening the food, moistening and lubricating, okay, and lubricating the food also, that mucus collect all the food particles, and it is made into that food. Now it is converted into a bolus, a mass, a round mass, just like laddu, a small laddu, like a small mass, that is called now it is bolus, bolus, uranda, bolus. இப்போ நம்ம என்ன பண்றோம், next bolus, அது வந்து என்ன பண்றோம், then next stage, that's all. Next stage, swallowing. So that is swallowing. So when swallowing, that food reaches through the, that goes to pharynx, and through the esophagus, okay, that reaches the stomach. As the swallowing is called, so deglutition, okay, that is swallowing. It is, listen, this is very important. It is that when that food goes through the esophagus, that wave-like movement, see here, it's beautifully, that is, that food passes, it is a wave-like movement, that wave-like movement, a contraction, that simultaneous contraction, relaxation. So that causes that gradually that food descends and reaches the stomach. So, okay, that wave-like movement is called peristalsis. That is called peristalsis. That wave-like that movement is called peristalsis. Peristalsis. Okay, so that is controlled by gastroesophageal, that sphincter, that is made up of that sphincter muscles. That sphincter muscles. So that okay, during the swallowing or the deglutition, okay, that food that passes that from the buccal cavity, that pharynx, from the pharynx through the esophagus, see, that slowly descends by the wave-like that movement, that is called the peristalsis. By the peristalsis wave that reaches the stomach. Yeah, that food, see, that reaches the stomach. Okay. So this is, that's all. Now this is the process takes place in the buccal cavity, that process of digestion in the buccal cavity. So after that that process completed, that is that swallowing, okay, or called deglutition, that reaches the stomach. Okay, that's all. And now mucus function, that salivary amylase, that digests the starch into maltose. 30% of the digestion takes place in the, that starch is converted into maltose in the buccal cavity. And one more, that is saliva also contains the enzyme, that is an antibacterial agent. It is called lysozyme, that kill any bacteria which were ingested along with the food, that kill the bacteria. Okay, that's all. Process of digestion in buccal cavity completed. Now you have seen that food reached the stomach. That food that is that bolus in the form of bolus that reaches the stomach. Now the food is in the stomach. See, listen, that reach. Okay, what is the process? What is the process of digestion? What is the digestive process in the stomach? Okay, and it is the stomach is this sac-like structure here, see, where the food is stored, that is lodged, remain 4 to 5 hours, subjective. So after reaching the food in the form of bolus, so that food it's remain in the stomach for 4 to 5 hours, right. Now the food is digested. So digested, how? So here that walls of the, already we have studied that walls of the stomach, it's namely gastric glands that secrete the gastric juice that contains the HCl and the enzymes, okay, enzymes like pepsin, renin, enzymes are there. See, listen, after reaching the stomach, see that walls of the stomach also that makes that alternate the contraction and relaxation. See the movement. Okay, this is called peristaltic movement. So, by the action of this peristaltic movement, that is and also that gastric juice, because of the action of the HCl and the enzymes and that food, okay, that is called churning process. Here that churns, so that enzymes mixes with the food and digestion takes place. Okay, and digestion takes place. So, listen, the bolus now, by the action of this peristaltic movement, that is the churning, okay, and action of the enzymes, now the food is converted into a creamy liquid. It is called a chyme. Okay, it is called a chyme. Or a creamy liquid. ஒரு creamy liquid, கூழ் மாதிரி, ஒரு பாயாசம் மாதிரி, அப்படியே என்ன பண்ணுது, அப்படியே ஒரு liquid, ஓகே, ஒரு creamy liquid-ஆ கன்வெர்ட் ஆகுது. ஓகே, ஒரு பாயாசம் மாதிரி, அந்த மாதிரி ஒரு கூழ் மாதிரி அப்படியே என்ன பண்ணுது, அப்படியே கன்வெர்ட் ஆகுது. ஓகே, அப்படியே வந்து now it is called chyme. Okay, by the peristaltic, that churning, that is called the churning. Okay, churning, that peristaltic, see the movement. Okay, so like this, that stomach, that is that moves, that peristaltic, that wave-like movements are produced. That is churning, that process. Okay, that grinds the food by the action of body temperature, HCl and enzyme, everything. Okay, and grinds, that food is converted into a creamy liquid. It is called a chyme. Okay, and you know what are that enzymatic action? Enzymatic action. So, you know that gastric juice, gastric gland secrete the gastric juice that contains, you know, that HCl and the proenzyme. HCl and the proenzyme. Proenzyme means inactive. Okay, inactive precursor. Inactive precursor. That is proenzymes. Okay, that proenzymes, it is pepsinogen. That proenzyme, name of the proenzyme is pepsinogen. Okay, that proenzyme is pepsinogen. That proenzyme pepsinogen. That pepsinogen, by the action of the HCl, that pepsinogen converted into that active pepsin. That active pepsin. So, with the help of the HCl, this is the function of the HCl. Okay, one of the functions that convert the proenzyme, inactive pepsinogen into pepsin, into active pepsin. Okay, what is the role of the pepsin? That pepsin act upon the protein. Listen, pepsin act upon the protein and convert the protein into proteases and peptones. Proteases and peptones converted into that further, that is proteins break into that is peptone. Okay, peptone and proteases. So, I said that enzyme, two enzyme, that is enzymes, that gastric juice contains what is that? Pepsinogen, that is a precursor. Just now I discussed pepsinogen, and another enzyme is renin. Okay, what is that? Enzymes, HCl, right? All secreted. That gastric juice consists of the enzymes, proenzyme, pepsinogen, that function, that pepsinogen converted into active pepsin by the HCl. So, that act upon the proteins and convert the protein into proteases and peptones, and another hormone is renin. Another hormone is renin. See, this is also the protein digesting enzyme. So, like the pepsin, this is also the protein digesting, but very particular, so that digests the milk protein, very particular, so that digests the milk protein, namely caseinogen, namely caseinogen, that digests the milk protein caseinogen into casein in the presence of calcium ions, very, very important. So, in the presence of calcium ions, that caseinogen, milk soluble milk protein caseinogen is converted into casein, that is a calcium casein compound, casein, curd, okay, curd, that is converted into curd. See, listen, this is very important. So, this renin enzyme which is highly secreted more in case of infant, baby, குழந்தைக்கு தான் இருக்கும். ஏன்னா குழந்தை தான் மில்க் ஃபீட் பண்ணும். நீங்கள் பார்த்துருக்கலாம், குழந்தை மில்க் சாப்பிடும், வாமிட் பண்ண அப்படியே கட்டி கட்டியாக இருக்கும், பார்த்துருக்கலாம் நீங்கள். ஓகே, அதான் தட் இஸ் வந்து என்னது, அதான் அந்த ரெனின், ஆக்ஷன் ஆஃப் த ரெனின், தட் மில்க் ப்ரோட்டீன் கேசின் பிரிசிபிட்டேட் ஆகிடும், வாமிட் பண்ணால் அப்படியே கட்டி கட்டியாக கேர்டாக வரும், கேர்ட், அதான் தட் இஸ் கேசின், தட் இஸ் தி ஆக்ஷன் ஆஃப் தி enzyme, namely renin. Okay, so this is more, that secretion is more, renin secretion is more in infant. When aging, that reduces. In adult, it may be absent. That renin, if secretes little quantities, very little quantities of the that renin is produced there, that this secretion of the renin that decrease when increased in aging, inversely proportional. Okay, when baby, that infant, that more renin, okay, renin secreted, it's more in case of infant. When that aging, when that's growing, that gradually decreases. In adult, it is absent. If may secrete, very little quantities are secreted. Okay. So, that the enzymes, proenzyme, this is only that the enzymes, there are two enzymes. Suppose the question is asked, name the enzyme present in the what is that gastric juice is the pepsin and renin, two enzymes, pepsin and renin. So, pepsin is secreted as the proenzyme pepsinogen, that is all. That pepsinogen is converted into active by the action of the HCl, that is secreted by the stomach, okay, gastric gland. That same that HCl convert that proenzyme into active form. Okay, then next, this is the frequently asked, what is the function of the HCl? Okay, what is the function of the HCl secreted by the stomach? Okay, that gastric juice contains, that HCl, what is the role? So, already that first it converts the proenzyme, that proenzyme pepsinogen into pepsin. Also, this is very important second. So, HCl, it provides the acidic environment that make the food acidic condition, So, in that acidic condition only, so that pepsin and renin, so that enzymes so will act efficiently. So, in order to that is that facilitate that enzymatic action, so that food, it should be in acidic nature. So, HCl make the food acidic, okay, that is optimum for the listen, it is optimum for pepsin. So, in the acidic condition only, so the action of the pepsin, the reaction of the pepsin will be more. So, it make the food acidic, and that HCl, that kills the bacteria, not only that activate the proenzyme and that make the medium food, medium here it is food, make the food acidic condition, also that what is that HCl kills the bacteria, kills the bacteria, so which is consumed along with the food. So, thus it avoids the petrifaction, okay, that is what is that further infection, okay, that it kills the bacteria. So, that is all. This is the that process, the digestive process, that enzymatic action that takes place in the stomach, and gastric juice contains that enzymes HCl, also that mucus and bicarbonates, also that mucus and bicarbonates, okay. What is the role? that bicarbonates and mucus that make that lubricate the food, also that mucous membrane, that mucus so that forms a layer, that forms the layer. So inside that forms the layer, that inner wall, of the stomach, that forms a layer, that is a protective layer. So that is what is the function, it protects the internal layer of the stomach from the action of the HCl and enzymes, okay, that protect that mucosal, that inner mucosal epithelium, that inner lining, that is the outermost, that is the innermost layer of that stomach, okay. So that mucus and bicarbonates, so that protect the inner lining of the stomach. So alright sir, that mucus it form a layer, mucus that form a layer, thus it protects and lubricate. How the bicarbonates, what is the role of bicarbonates, how the bicarbonates helpful in the protection of the that internal layer of the stomach? You know that bicarbonates that neutralizes the acid, that neutralizes the HCl. So that HCl, that action, that power will be decreased, okay, that HCl power is decreased by the action of the bicarbonate, okay. That bicarbonate it neutralizes already acid, this is the base salt, that neutralize the acid. So thus that bicarbonates and mucus which protects the so inner that layer of that stomach, okay. So that's all. So far that digestion in the buccal cavity and the stomach. Then next is, so after the food is digested by the action of the gastric juices, the food is churned, so and become a semi-solid substance known as chyme. So now the chyme, so gradually, slowly from stomach to that small intestine that reaches. So now we are going to discuss so what are the digestive process takes place in the small intestine. The small intestine, which is the major part, it's not only that small intestine that not only meant for digestion, also the absorption of food also takes place. So here that food is digested by the major glands, namely the liver, pancreas and intestinal juices. So the secretion of all these glands act upon the food. So the 100% of the digestion that takes place in the small intestine. So that enzymes and also that movement, okay, like that wave-like movements here, that small intestine. So in the small intestine, that wave-like movements are produced. So that mixes the food and enzymes, so that facilitate the digestion, right? And the first gland, as I said, three major glands, namely that liver, pancreas and the intestinal gland pour the secretion, so that act upon the food. That first is the liver. So you know that is the largest gland that secrete the bile juice. So what is the composition of the bile juice? That bile juice, it contains bile pigments The bile pigments, which are the breakdown product of the RBCs, okay, the bilirubin, biliverdin and bile salt, okay, bile salt, cholesterol and phospholipids. So, these are the composition, these things are found in the bile juice and no enzymes. So, in bile juice no any enzymes, only it contains the cholesterol, bile salt, bile pigments and phospholipids. So, what is the function of the bile? So, the bile which emulsify that salt, that bile salt which emulsifies the fat. So, emulsification, it is saponification. So, during that process, that insoluble fat we consumed, fat, korupu, lipid. So, that which is insoluble, that fatty substances are insoluble. That insoluble fats which are converted into soluble form by that bile salt. That process is called, technically called, it is emulsification. So, that reduces the surface tension. As a result of these, that fat substances are converted into small fat globules, so that lipases. So, because of that, the lipases act upon that lipids. So, lipid is a fat, that enzyme which act upon the fat and digest is called lipases. So, because of this emulsification, so that bile activates that facilitate that action of the lipases, so that the lipase act freely, so on the lipids, so easily that digest the lipid. So, these are the functions of the bile. So, mainly the emulsification. Without emulsification, so that lipases cannot act on the lipid, okay, and unable to digest. So, so that the biles which activate the lipase, so it is an enzyme that which act on the lipid. The enzyme which act on the lipid is called lipase. So, that bile that facilitate that activate the lipase to digest the lipid, right, and convert into small fragments, fat globules, and emulsify the fat. So, these are the functions of the bile. Separate the 3 marks question. This is the separate 3 marks question. Write the functions of the bile. So, what are the functions of the bile? Okay, that is all, that components and functions of bile over. So, the next is the pancreatic juice. So, you know that is it is a leaf shaped yellowish gland which is found below the stomach, is called pancreas. That pancreas secrete pancreatic juice, pancreatic juice. So, that pancreatic juice contains the enzymes like trypsinogen, chymotrypsinogen, carboxypeptidase, pancreatic amylase, pancreatic lipase and nuclease. So, these are the enzymes which are found in the pancreatic juice. That is the pancreatic juice is nothing but that juice secreted by the pancreas that contain these enzymes: trypsinogen, chymotrypsinogen, carboxypeptidase, right, pancreatic amylase, pancreatic lipase and nucleases. That 6 enzymes. So, what are the role of each and every enzyme? See, that trypsin and chymotrypsin, the trypsinogen and chymotrypsinogen are the inactive precursor. See, the trypsinogen and chymotrypsinogen are the precursor. So, inactive. It's secreted in the precursor or inactive or non-functional state. Okay, then which is the trypsinogen and chymotrypsinogen are converted into active form. So, which are converted into active. Trypsinogen is converted into trypsin. Trypsinogen is converted into trypsin and chymotrypsinogen is converted into chymotrypsin. Okay, chymotrypsin, trypsinogen, it is a precursor which is converted into trypsin. Chymotrypsinogen is converted into, this is precursor which is converted into chymotrypsin by a enzyme. So, by an enzyme it is called enterokinase, very important MCQ. That precursor enzymes trypsinogen and chymotrypsinogen are converted into the trypsin and chymotrypsin by the enzyme known as enterokinase, which is secreted by the intestinal gland. That intestinal mucosa, it's nothing but the innermost layer of that small intestine. You can write that enterokinase, it is an enzyme which activates the proenzyme into that active form, which are secreted by, that enterokinase is secreted by the gastric, that is intestinal gland, and that produces one of the enzymes, enterokinase, that activates. This is important. And what is the role of the trypsin and chymotrypsin? So, the trypsin, it is both the trypsin and chymotrypsin or the proteolytic enzyme, that is both the enzymes digest the protein. See that the trypsin act upon the protein and convert the protein into polypeptides and peptones. So, here that chymotrypsin, so that also, as I said earlier, it is also the proteolytic enzyme and convert the protein into polypeptides and peptones same, but what is that difference? So, that which act upon the chymotrypsin that act upon the specific amino acids, that specific amino acids from the protein that remove the specific amino acids. So, what is amino acids? Amino acids are the building blocks of the protein. Building blocks of the protein. It's a unit, monomer. Okay, many amino acids join by the peptide bond. That is many amino acid plus amino acids. Many amino acids join by the peptide bond and form a polypeptide chain. Polypeptide chain, you know, that is different types of amino acids, 20 different types of amino acids that join and form the protein. And this chymotrypsin, so that act upon some specific amino acids, okay, that breaks certain specific amino acids, so that that polypeptide chain, a protein is technically it is called also known as a protein or polypeptide chain. Amino acid plus amino acids, peptide, that is by the peptide bond that form a polypeptide, long chain like structure. Okay, and that from the polypeptide chain that breaks certain specific amino acids, so that it is break into polypeptides and peptones. Polypeptide and peptones, both are the proteolytic but specific, that chymotrypsin which act upon the specific amino acids and remove that amino acid from the polypeptide chain. Okay, the next enzyme is pancreatic amylase. Pancreatic amylase, see, listen, it is starch digesting, that act upon the starch. So, here glycogen and starch. It's glycogen, it is like a starch. Okay, it is both are polysaccharides. That what is the glycogen? That if it is that polysaccharide stored in the plant, that are called the starch. Suppose same in our body, that same that polysaccharide, if it is stored in our body, that is called glycogen. So, this simply that the glycogen and starch are the polysaccharide. So, that pancreatic amylase act upon the that polysaccharides, namely glycogen and starch, that convert into maltose. So, listen, so these both are the polysaccharide. Both are the polysaccharides. So, these polysaccharides are converted into that maltose. Maltose are the disaccharide, which are the disaccharide. Okay, disaccharide. So, that polysaccharide is it's a break into imagine a long chalk piece. It's a polysaccharide, that is you break into two pieces. So, that is maltose, which is a disaccharide. The disaccharide further split into monosaccharides, right. So, the pancreatic amylase act upon the polysaccharides, namely glycogen and starch, then convert into maltose. And similarly, the next is the lipase, you know, that lipid, that enzyme which act upon the digest the lipid is called lipase. So, lipase act upon already that fat which are, you know, which are in emulsified condition. So, that lipase act upon the emulsified fat and convert into free fatty acid and monoglycerides. Free fatty acids and monoglycerides. So, that end product of the fat is fatty acid, that lipid is the end product, that final resultant product of the lipase is fatty acid and glycerol, that is the end product. Now, it is a free fatty acid and monoglycerides. This is the one of the end product. This need not be further, so break into pieces, but that monoglycerides further hydrolyzed and form the fatty acid and glycerol. Fatty acid and glycerol. So, thus that fat is fully digested, lipid, the fat is fully digested here. That lipase act upon the emulsified and convert the free fatty acid and monoglycerides. This is the complex substance. These further, that monoglycerides further hydrolyzed. Hydrolyzed means digested, further break down and produces fatty acid plus glycerol. This is the end product. So, these need not be further divided, okay, digested. This is the end product of the lipid digestion. And nucleases, that nucleic acids, DNA and RNAs are called nucleic acids. That nucleases which act upon the nucleic acid, namely DNA and RNA, and convert into nucleotide and nucleoside. Nucleoside and nucleotide. That nucleases act upon the nucleic acids, namely DNAs and RNAs, and convert into nucleotide and nucleoside. That structure, this is the structure. That is a nucleoside. Nucleotide, and that is DNA is called polynucleotide. DNA is called, it is a polynucleotide. DNA and RNA are polynucleotide. That means many nucleotide. That nucleotides are monomers. Many monomeric nucleotides join and form a long chain. Polynucleotide. It's nothing but DNA. DNA is many nucleotides connect like a chain. Many links interconnect, long chain. So, அந்த மாதிரி nucleotide அது ஒரு chain formation ஆயிருக்கும். அப்ப nucleotide அப்படின்னா என்ன அப்படின்னு பார்த்தீங்கன்னா ஒரு நைட்ரோஜினஸ் பேஸ், அது ஸ்ட்ரக்சர். ஒரு நைட்ரோஜினஸ் பேஸு, அதில் அடினைன் தைமின் அது மாதிரி ஏதாவது ஒரு நைட்ரோஜினஸ் பேஸ் இருக்கும். அது கூட ஒரு சுகர், பென்டோ சுகர், ஓகே, ஒரு சுகர் ப்ளஸ் ஒரு பாஸ்பேட் மாலிகுல் இருக்கும். ஓகே, ஸோ இவங்க இந்த 3 பேரும் தட் இஸ் நைட்ரோஜினஸ் பேஸ், அடினைன் தைமின், குவானைன், சைட்டோசைன், any one of the nitrogenous base and sugar, phosphate. So, இவங்க தான் nucleotide. So, இது அப்படியே என்ன ஆயிருக்கும், repeated. இது அப்படியே repeat ஆயிருக்கும். Many nucleotides form a long chain. அதான் DNA and RNA. இப்போ என்ன பண்ணணும்னு கேட்டிங்கன்னா, listen, இந்த 3யும் ஜாயின் பண்ணி இருக்கிறது தான் நியூக்ளியோடைடு. அப்போ என்ன பண்ணணும்னு கேட்டிங்கன்னா, that is that nucleases பார்த்தீங்கன்னா that is இதை பிரேக் பண்ணும். ஸோ இந்த மாதிரி நியூக்ளியோடைடாகவும் நியூக்ளியோசைடாகவும் பிரேக் பண்ணும். அப்போ இதான் நியூக்ளியோடைடு. நியூக்ளியோசைடுனா என்ன? இதுலேருந்து இந்த பாஸ்பேட்டை மட்டும் ரிமூவ் பண்ணுங்க. பாஸ்பேட்டை மட்டும் ரிமூவ் பண்ணுங்க. அப்போ என்ன இருக்கும்? ஒரு சுகர் நைட்ரோஜனஸ் பேஸ் இருக்கும். ஸோ இந்த யூனிட்டுக்கு பேர் தான். ஸோ ஓகே, திஸ் யூனிட் that is called nucleosidases. நியூக்ளியோடைடு, இது நியூக்ளியோடைடு அண்ட் இது நியூக்ளியோசைடு. ஸோ ஃபர்தர் இது பிரேக் ஆகும். அது என்னன்னு பார்க்கலாம் நம்ம. ஓகே, nucleases simply that act upon the nucleic acid that convert the nucleic acid into nucleotides and nucleosides. So for your understanding I am telling, this is additional. Okay, this is additional. For your understanding I am introducing this. Nucleotide na enna, nucleoside na enna, that is the units of the DNAs. Nucleotide. So right, and so far we have discussed the function of the pancreas. Okay, the pancreatic juice. The next is the intestinal juice, which is also known as succus entericus. One mark. That intestinal walls of the intestine also possess a group of secretory cells, glandular cells, that are called crypts of Leiberkuhn, that secrete the juice that is called intestinal juice or succus entericus, which is also known as succus entericus. So what are the enzymes? What are the enzymes which are found in the intestinal juice or succus entericus? Maltase, see listen, very important. Maltase, lactase, sucrase, peptidase, listen carefully, lipases, nucleotidases and nucleosidases. Nucleotidases and nucleosidases. So these are all the enzymes. So these are all the enzymes which are present in the intestinal juice or succus entericus. Okay, succus entericus. What are the enzymes? Maltase, lactase, sucrase, peptidase, lipases and nucleotidases and nucleosidases. So now let us discuss the function of the each and every enzyme. Maltase, sucrase, lactase. Maltase, sucrase, lactase. இப்போ நம்ம என்ன ஆயிருக்குது நம்ம பார்த்தோம். What is that? Pancreatic juice, அதில் உள்ள pancreatic amylase, so that break digest the starch into maltose. நான் சொன்னேன் starch into maltose. starch என்றது வந்து என்ன? It is polysaccharide. polysaccharide disaccharide break பண்ணிருக்கு. அதில் பாருங்களேன், அந்த டைசாக்ரைட்ஸில் maltose, sucrose, lactose. There are three types of disaccharides இருக்குது. maltose, sucrose, lactose. All the three are the disaccharide. என்ன டிஃபரன்ஸ் சார்? maltose, sucrose, lactoseனா என்ன? இப்போ நார்மலாக வந்து பார்த்தீங்கன்னா maltose வந்து பார்த்தீங்கன்னா கிரைன்ஸில் இருக்குது, சீரியல்ஸ், ரைஸ் மாதிரி சீரியல்ஸில் இருக்கிற அந்த ஸ்டார்ச் வந்து டைஜஸ்ட் ஆனால் மால்டோஸில் ஆகும். That maltose is the disaccharide, so present in the cereals. Okay, cereal, rice மாதிரி அதான். அதில் இருக்கிறது maltose. Sucrose, can sugar. That is அதுவும் ஒரு disaccharide தான், but name different. Sucrose, it is the disaccharide present in the can sugar, சக்கரை. Lactose, it is the disaccharide present in the milk. மில்க்ல இருக்கிற சுகர் வந்து lactose, lacto milk. Lactose, sucrose and maltose. So these all the three are the disaccharide. So these are produced by the action of the pancreatic amylase. So pancreatic amylase convert the polysaccharide into respective disaccharides. இப்போ maltose further it should convert into monosaccharides, polysaccharide, so converted into polysaccharide disaccharide further divided into, that is breakdown into or digested into monosaccharides. அப்போ these are the disaccharides. Maltose, sucrose, lactose are disaccharides. Maltose, how it is digested? Which maltase act upon the maltose and convert the maltose into glucose and glucose. Glucose plus glucose break பண்ணுது. That is the disaccharide maltose contain glucose. அப்போ அது 2 glucose, the di. அப்போ அந்த maltose is break into, digested into glucose plus glucose. Next is sucrose, cane sugar. So that sucrase act upon the sucrose, ASC, okay, that suffix ASC, அது end ஆச்சுன்னா அது name of the enzyme. அப்போ sucrose, that இது substrate, the enzyme is sucrase act upon the sucrose and convert into glucose and fructose. Then lactase, similarly that lactase act, these are the enzymes, these are the substrate, that lactase act upon the lactose and convert into glucose and galactose. See, listen, that glucose, fructose, galactose, they are the what are the isomers, right? Same molecular formula but different structural formula. See now the function of the that intestinal juice, namely maltase, sucrase, lactase over. And next is the peptidase act upon the protein. I said already that the peptide bonds that connect the amino acids, that polypeptide, the peptidase that break that peptide bond and convert the protein into amino acids. நம்ம பார்த்திருக்கோம். இப்போ வந்து என்னவா இருக்குது? Protein is not completely digested. அதுவும் புரோட்டீன் எங்க ஸ்டொமக்ல வந்து என்னவா இருக்குது? that is polypeptides, peptone, proteases, அந்த மாதிரி சின்னதா இருக்கு. that further, so that dipeptide, tripeptide, polypeptide, okay, அவங்கெல்லாம் என்ன பண்ணுவாங்க? இப்போ not protein வந்து ஒரு செமி டைஜஸ்டட் கண்டிஷன்ல இருக்குது. dipeptide, tripeptide, polypeptide, that condition. so அவங்க மேல என்ன பண்ணுவாங்க? that peptidase, okay, peptidase act upon the semi digested protein and convert into amino acids and finally nucleases நாங்க சொன்னோம். nucleases அவங்க என்ன பண்ணுவாங்க? நியூக்ளிக் ஆசிடை வந்து பாத்தீங்கன்னா நியூக்ளியோடைடு அண்ட் நியூக்ளியோசைடா பண்ணுவாங்க. நியூக்ளியோடைடுன்னா நான் சொன்னேன், தட் இஸ் நைட்ரோஜனஸ் பேஸ், சுகர், பாஸ்பேட், அந்த ஒரு கம்ப்ளீட் யூனிட் தான் நியூக்ளியோடைடு. இப்போ என்ன பண்ணு கேட்டீங்கன்னா நியூக்ளியஸ்லயே there are 2 type of nucleases இருக்குது. நியூக்ளியோடைடேஸ் அண்ட் நியூக்ளியோசைடேஸ். நியூக்ளியோடைடேஸ் அண்ட் நியூக்ளியோசைடேஸ்னு இருக்குது. அது என்ன நியூக்ளியோடைடேஸ்? அப்ப நியூக்ளியோடைடு இருக்கு பாருங்க, அந்த 3, all the 3 இருக்கு பாருங்க, நைட்ரோஜனஸ் பேஸ், சுகர், பாஸ்பேட். okay, that nucleotidase act upon the nucleotide வந்து என்னது? okay, nucleotide the full unit, that nucleotidase act upon the nucleotide and break into, listen, nucleoside and phosphoric acid P. நான் சொன்ன பாருங்க, அந்த P எடுத்ததுக்கு அப்புறம் இருக்கும் பாருங்க 2 sugar and nitrogenous base, that is called nucleoside. அதுவும் ஃபர்தரா வந்து என்ன பண்ணும்? ஃபர்தரா it should be further digested. அப்ப அது that is nucleoside. nucleoside மேல யார் ஆக்ட் பண்ணுவாங்க? that enzyme nucleosidase and break that nucleoside into nitrogenous base and sugar. so that nitric, that is what is that the units DNA and RNA, Okay. Nucleic acids also fully digested. And see, so in the small intestine, that is by the action of the pancreatic enzyme, maybe fatty substances, lipids, if anything is there, so that are further the diglycerides and monoglycerides are further digested by the lipase, and finally that convert into all the lipids are converted into, if anything, any remaining fatty substances are there, any fat are there, so that are digested by the lipases, and so convert all the lipid or fat into the fatty acid and so glycerol. So now all the that food we eat is completely digested, digestion process get over. See, listen. So this is the end product of the carbohydrate, that is starch, glucose, fructose and galactose. All the protein or break into amino acids. This is the end product. Okay. And nucleic acids are converted into what is the sugars, nitrogenous base and phosphate separated, and fatty substances or break down into fatty acid and glycerol. So thus the 100% of the digestion get over. Okay. And the next process, I said, so these are the end product of the digestion. So that food is completely digested, that breakdown, that complex molecules are so break down into simpler. So the respective that monomers, okay, which are converted, that is the digestion, that chemical digestion, that enzymatic action, so that are completed. Then next is the absorption. The next process is the absorption, that food, so which are absorbed, okay, the food which is absorbed by the that villi, that intestine, small intestine also possess some finger-like projections, so that are called intestinal villi, so that absorbs undigested fecal matter, that is undigested waste. Okay. So undigested waste materials, so which are that is transported to the large intestine and then temporarily stored in the rectum. From rectum, so that undigested fecal that is waste matters are sent out in the form of feces, in the feces. So I hope you all so understood the process of so digestion that takes place